piece. So let's give it up for Dr. Hazan. Thank you so much for presenting.
Thank you, Addy. As you know, my name is Hananel Hazan, and I'm from the Allen Discovery Center in Tufts University. I would like to talk to you about two. Oh, my computer's stuck. That's fun. I would like to talk to you about two different possibilities that you can use biology in our environment today that I think are well-deserved more attention and more people working on them. So the first is that, as you can know already, the computational growth of very variation algorithms like ChatGPT, large language models, diffusion models, all of those programs require huge amount of computational resources. And without computational resources, also demands for electricity and resources, which is tremendously can overwhelm our infrastructure, which we should somehow going to hopefully develop in biology. Another field, as Dr. Field just talked about, is a medical intervention using techniques that come from engineering. The Levin Lab show amazing results using models that can be manipulate and sometimes even treat some biological problems that we currently have. So let's start with the computational resources. We know if you plot, and I know that it's already by now in the computational field, this is an old diagram. We can see that the growth of the computational resources needed today is exponentially growing. Every year we are doubling the needs of our algorithms, and every year we are, as sequencers, we need more computational power, that need more electricity, that need more resources. Everything in that chain require more and more process. But there is some limitation that we can grow on. For example, the shrinking size of the silicon chip, there is some limitation that we eventually going to reach it. The electricity demand that we will need in order to get to that computational resources will eventually reach. So what can we do? And one option is to look no less than biology. The rat brain take very, very small amount of computational resources. It's about 0.04 watt. Our brain require about 20 watts. And compared to that, a very old algorithm by now, AlphaZero, require 3,000 watts. And I'm not talking about even ChatGPT and all of those algorithms that are really, really in demand. So in a way, we can look for the computational point of view in biology cells. So even if we are talking about the computers today, or the neural network of today, the neural network of today are reminiscence or the predecessors of the models that start in 1957 in the called Presotron. Well, that algorithm was a discrete, as is today. We didn't change much. The only way that we change is that growing not one perceptron. A large language model contain billion of parameters, which mean millions of neurons that connect between them. And that kind of algorithm is very different of what biology use. Biology use, and in cells, in neurons, they use an analog. And the complication or the on the complexity of organism, one simple cell is very, very much very complicated compared to what we use to describe a neuron. This is an illustration of a neuron, as to claim that this neuron is resembling to the human brain is probably similar to claiming that Homer Simpson is a representation of human body. So just give you some illustration. This is how neurons look like under the microscope. This is how they operate, and they flashing. They doing their activity is about a flash or not flash, or spike or not spike, and this is very appealing to computer science that are operating in binary world, but not like computers or algorithm in the deep learning. The neurons under the microscope, you can see them growing and try to reconnect to other neurons and the neighbors. So in a way, they try to change their algorithms and change the architecture. And they growing or manipulating the way that they transmitting information throughout the tissue. A little bit more complicated than that. This is a cortex of a rat brain under the microscope that been manipulated with some monitors, proteins that gave him different colors. So what you can see here, the yellowish and the yellowish object that you see running around here is some cells that call glia and astrocytes. And astrocytes are moving cells. You can see them running away. They are usually considered to be cleaning the leftover and the stuff that are left from activity of neurons. And the green neurons are the excitatory neurons, and the red neurons are inhibitory neurons, which is really, really complicated compared to our simplified version of neurons. But in a way, we can use them in order to understand how also human brain work or nervous system work, and try to use them as a computational devices that can help us to process much more information in the future. How can we do something like that? We use something called MEA, multi-electrode arrays, and those are the cells that you can, the Petri dish here that you can see here, and there are cells inside, and this is how they look like under microscope. You can see the cells around here, and those are dots are electrodes that are sensing the information or the activity, and also manipulating and stimulating the activity. So in this way, we can transmit information to the tissue and encode the environment and to see how they react. So a team of my old laboratory in the Technion used this already in 2008 to use a robot that send in sensors to a Petri dish of cortical neurons. And that cortical neurons had a decision to make how to avoid and go forward and avoiding obstacles on the platform. And that wasn't as simple, but very, very rewarding, because you can do that with less than 10 neurons on the Petri dish, which is amazing compared to the vast of computational resources that you would need to do in order to create an algorithm that will solve it. So since you are bioelectricities or biohackathon, I don't want you currently now using biology to work on your project, but one of the options is to do a simulation. One of the simulations that we have is BindsNet that use the information like an Atari game and try to connect it to a spiking neural network in order to manipulate and control the algorithm in the environment. And this is an example how we use a spiky neural network to play a ping pong game, which at that case was very much successful compared to the artificial neural network that been trained in the similar way, but was less successful. So if you want to try, this is one of the environment that we use in our simulation. Let's jump ahead and to the medical point of view and not only the computational power. And this will be the second subject. And in the medical point of view, or in the biological point of view, as a computer scientist, I'm seeing cells are very interesting creatures. In a way, each cell starts from the same ancestor. They all start from one sperm cell that been divided and modulate and still divided and clone. So it means that every cells in your body and our body contain the same instruction set. In computer science, we will call it the same programming. But each one of those cells are differentiate and specialize to a different method. Some of the cells become skin cells, some of the cells become liver cells, and each one of them operate on their environment. And this is something very, very interesting to work on, because what kind of information or what kind of program you can create in order to take in account all of these complexities that we have in the human brain or human body or in every creature that you have. So again, let's start with some simulation. We can simulate each cells in a series of capacitors, resistance, and inductors. And in a way, we can create a simulation of how biological tissue look like or will behave in a certain conditions. And for that, we build another simulator that called BETSI simulator, a biological electric tissue simulator. And Alexis, one of the members in the lab, use this simulator to do something that I think it's amazing in terms of way of manipulating biology. They had experiment, and in this experiment, they use a frog oocytes, and you can see it here in the screen, and you can see the face of that frog in that moment. So in this development point, you see this is from the beginning. This development point, the tissue in the embryo being divided to two parts, and this part is the cleavage where it's being decided what cells going left and what cells going right. And once that happens, the brain of the tadpole being developed. And this is a normal tadpole, nothing happened to it, it's fine, normal. However, if during this growth, you expose the tadpole to a nicotine, what we'll see is that the control develop a normal brain, but the tadpole that exposed to nicotine did not develop a normal brain. And in that way, that tadpole was very defected, did not survive, and died very easily. So the question is, how can we manipulate the development of the cells, even if the tadpole being exposed to nicotine? How can we counteract the effect? So Pi, one of the senior members in the lab, measured the activity and the voltage of each one of the cells around the face of the embryo and discover that there is sort of a cleavage in the control in the normal frog cells that have that kind of cleavage that we can see it here. And that cleavage is being exposed in a level of voltage in the cells in that area here. And once the nicotine is in the system and that tadpole exists with the nicotine, that cleavage will not be developed strong enough in order to create the right division. So the question was how to find the right drug in order to manipulate this or to counteract this kind of effect. And Alexis tried to simulate everything in the simulation in Betsy. And after a lot of experiments where she did a very manual job in order to find the right compound, in that compound we abbreviated in IT here, which been introduced after the exposure to nicotine, and those tadpoles succeed to create the right cleavage, and the brain of the tadpole when formed pretty normally. So in the way to create that kind of to find this intervention, we're able only because we have the simulation to do that, because once we have the simulation, it's very easy to restart the experiment and start it in different conditions. It's very hard to do that in biology. Every one of those experiments, you need to grow the tadpole from scratch, wait about 3 months until it grows, until it exposed, and until you can get a reading from it. So to do experiments while one trial will take you 3 months will be enormous overtake. And there where simulation can take over by creating the condition and running many simulation, because we have computer powers in order to find the right agent, the right intervention to have that kind of correction. So in my work, I'm using the same simulation, and I'm trying to take the simpler way of parameters in order to find a program or a pipeline where a biologist can come to me and say, I would like to create this kind of a pattern. What I might need to do in order to create this kind of pattern? What kind of intervention I need to use in order to create a pattern? So I started with a simple solution, with a simple tissue, where I have roughly 6 parameters, which each one of the ranging range values. And you can easily see that once you add more and more parameters, the combinatorial space of the possibilities are growing exponentially. So in a way, to do that kind of search, we need more sophisticated tool beside just randomly pick a parameter and hope for the best. In a way, we can look at the parameter space, and this is only illusion, where we are somewhere in this parameter space and we want to achieve this target. And in order to not go in a random search, we need a way to understand the parameter space in order to find our way directly to the target. And that's where come a heuristic algorithm. Heuristic algorithm have many flavors, are many ways. Some of them can, and the one that we are very fond of them is a genetic algorithm, which is a very easy and simple algorithm, but it's really rely on a lot of guessing and randomness. So in the Levin lab, we develop a deep learning based on diffusion model, similar to the models that exist in creating our images on the platform like OpenAI, where we have the parameter space being explored using the algorithm, and the algorithm able to predict where the right coordination of the target is located. And we compare this algorithm to the regular genetic algorithm, which you can see here. In our algorithm, we are able to have multiple targets, and in the genetic algorithm is less way of finding multiple targets. We also have able to changing on the fly, meaning if the environment change, the algorithm itself learn and adapt to the new situation, which the regular heuristic algorithm that used today is not able to adapt. In this case, the target is find the green, and the genetic algorithm is not able to find the green after it gone and changed direction. And another thing that we can do is manipulate the search in a way that we can say, in a way of reinforcement learning, we can manipulate the search to say, we would like, for example, that the lunar lander that just landing here will not activate his thruster so much. Why? Because we need to consume energy. And this is very hard to do in a regular genetic algorithm. In our algorithm, it's much faster and able to sustain adaptation to a condition that we forcing on. So let's go back to the frog phase. We would like to find a parameter that once we start with a regular tissue, the regular tissue will converge eventually to be similar to our control or to our target, which is the control phase, the frog phase with the cleavage here. And this is one of the parameters that we found in our search, which what you see here, you see about 600 cells. Each one of them behave as a regular cells in the frog that have the same ion channels, the same gap junction, the same conductances, and we just need to find the right parameters in order to initiate the activity. So in a way, we're getting to the same pattern as we wanted from the beginning. And this is one of the results that we came up with. And another way of a cell that have a different voltage activity or voltage dynamic in different tissue. An example that we have is if, for example, the biologist would like to say, I would like to have a cells that eventually will create a smiley face. So this is our target, and we let the algorithm to find the right parameter that will end up as a smiley. And eventually, after a long experiments, we find the right combination how we should use what kind of concentration of ion channels concentration or drugs manipulation we need in order to manipulate the tissue to reach to the desired target, like this one is the smiley. Another question that we ask, again, because we are so much computer scientists, is there a way to find a combination that is not affect because of shape or number of tissue? And apparently it was very hard decision or hard search, but we did find a way to find an intervention that can work in many combination of cells that are in a different combination of arranging of cells, number of cells, or the shape of the cells. Another option that we have is a tissue that sustain a memory, meaning we stimulate it here at the beginning. We have it stimulate. Where is my mouse? In this figure, in the right figure, you can see the stimulation begin, and then the activity of all of the voltage activity of all of the tissue sustain a higher voltage. Another option that we had, if you know the Game of Life by Conway, Game of Life, so this is one of the games that we create, or glider, and we can find the same sort of combination in a tissue that creates the Game of Life in that tissue. Another method or parameter that we want to achieve is achieve similar to linear. Another simulation that we run, they have a simulation of creatures that create a mathematical creature. We want to achieve similar way and in our tissue. And again, we ran the parameters search to find the right parameters, and that's what it comes with. And some interesting shapes that you can see. Or cardiac cells, and this is one of the experiments that create cells that are fluctuating and operating like clocks, so they're oscillating. Again, this is something unique in those simulations, which you can see there are about 1,200 cells in those tissue, which each one of those cells contain the same algorithm and the same configuration of any other cells in the system. But as you can see, as a combination of them, as a dynamic activity, we can get different activation patterns, which is what we can see in biology, in biology scenario. And the last example is symmetry and asymmetry breaking, which you can see that the same cells, the same tissue create this symmetry activity, almost copy of the left and right tissue or left and right tissue. Again, creating a dynamic activity in that tissue is very hard to predict both in computational and in biological scenario. And with that, I would like to thank you for your attention and thank you for my collaborators.
Awesome. I'm going to assume there are maybe a couple of questions.
So regarding the first half of the presentation, how is that related to what Elon Musk is doing with Neuralink?
This is very related. What Elon Musk doing is intervention inside the brain already, which is different than simulation, because in simulation the situation is very hard. When you use biology is 10 times or 1,000 times harder than regular cells. But in a way, the simulation can help this kind of experiment, like what Neuralink doing, to predict where to stimulate, how to stimulate. Because you remember, when they do the experiment on human brain, you don't have a second trial. It's a human brain that we are talking. We don't want to guess and test. on a human. So before you do something on human, you should do that first of all on simulation.
What are the underlying assumptions of your simulated models?
That's a good question. Every simulation have a reduction, doing some kind of reduction. Every simulation create a simplified way of starting point, ion channels, and many other simulations. So the simulation can be only good as the data we created. That's the reason we are working with a lot of biologists in order to be near where the ground, the truth ground exists. So we try to simulate as much as what we need from biologists and not also bog down the level of complexity that the computer can do. For example, when you do a simulation, how low you can go in the way of simulating, how low you would like to simulate. Would you like to simulate the atom level, the moleculars, the ion channels interaction? Each one of those are different questions that we need to ask ourselves and depend on the target of the aim of the research. We find the Goldilocks where we need less compromise on parameters. So to your answer, depend on the task.
Hi. At some point at the beginning, you were saying a good idea would be to have a spiking neural network to play a game. And
Yes.
I want to know if you see an advantage of doing that instead of artificial neural networks. And my second question is, do you feel that adding more biophysical properties like ionic currents or synapticity would be helpful for a test like that?
Also, a wonderful question. So, the reason I love spiking neural network is because although it's hard to simulate them and you need a lot of computational resources to simulate them, in electronics it's very easy to use spiking neurons. There is many materials today that are cheap, reliable, and cost-effective to create neurons or spiking neurons in electronics. And once you use that kind of electronics, in order to simulate ChatGPT on the same computational resource or the same size of network, the spiking neural network will take less than 10% of the computational or the electricity resources that the artificial neural network will use today. So the reason is to use spiking is not only ecological or cost effective, is also once we succeeded to establish how to learn in using spiking neural network, we would able to really really a huge jump to an environment where the computational resources are at least 1,000 times stronger than what we can do with the same electricity demand.
Thank you, Dr. Hazan. I was actually going to ask if you don't mind giving a couple words of inspiration or motivation as people are trying to lock down on their ideas for the night, because they do have to submit this tomorrow. And we have a mix of people in biology, computer science, engineering, some math and physics people, I believe. So if there was anything you could kind of say that would really get them thinking out of the box, such as yourself, are there any things that sort of come to mind?
Ignore whatever other people say to you that it's not possible. Do that.
Does that include me and you?
Especially me and you.
Perfect. All right, last round, no more questions? Michael? Yeah.
So I just thought of a new neural network, or I don't know if it's a new neural network, and then I have to edit that, during your talk. What about one where like a neuron is able to look at a bunch of neurons in the previous layer, a finite list of them, and does a weighted sum of them, and depending on the resulting analog value, it gives a binary output. Would that be more similar to like a model of neurons in the brain?
It's very similar to the way that neuron working in the brain, but in the way that also neuron working in the brain are one caveat that I didn't show here in this presentation, that our brain doesn't use backpropagation algorithm, which is the main engine beyond all of the models that been used today in artificial neural network. There are many requirements or constraints that biology have that backpropagation algorithm is not able to work in those constraints. So the big question is how neuron learn, how neuron adapt, what is nature come with genes, and one is nurture. We learn coming from learning the environment. And that's where our need you to think about new ideas.
Awesome. Thank you. Give it up for Dr. Hazan.
So with that, we just thank you so much for your time and for a very fascinating topic. So have a great evening. We're going to sign off the Zoom and get down to business.
Thank you. Good luck.
Night.